Welcome back to the Science Channel podcast, where we discuss the coolest scientific papers and preprints of our day.
Great to be here again.
And hey, if you're enjoying these deep dives into the science shaping our understanding of the world, please do hit that like button and subscribe. It really does help the channel grow.
Absolutely. Every click helps.
So today, we're tackling something pretty fundamental, I think. It has surprisingly wide-ranging implications.
It really does. We're talking about mitochondria, the powerhouses in our cells.
Right, the energy generators.
Exactly. But we're looking at their role in inflammation and immunity. And it's a story that, it stretches back billions of years.
Billions. That's hard to wrap your head around sometimes. These tiny things inside us have such an epic history.
It's incredible.
They weren't originally part of us at all, were they? They were separate organisms.
That's the prevailing theory, yes. The idea is they were once free-living bacteria, and way back when, some ancient cell engulfed one.
Engulfed it, but didn't, digest it.
Precisely. Instead of being destroyed, it sort of stuck around, formed a partnership, a symbiosis.
Endosymbiosis, right, inside.
Exactly, endosymbiosis. And over vast amounts of time, this became permanent. They became part of the cellular machinery.
But they still carry echoes of that bacterial past.
That's the crucial point. And it's this ancient origin that still shapes how our bodies work, especially our immune system.
Okay, so this former bacterium, now part of us, but with lingering bacterial traits. And the paper we're diving into today, let's see, it's a break in mitochondrial endosymbiosis as a basis for inflammatory diseases. That's by Michael Murphy and Luke O'Neill, published in Nature.
A really top-tier journal, and a very provocative title.
It is. It suggests these bacterial leftovers can actually cause problems, right? Like our immune system gets confused.
That's the core idea. Our cells pick up on these, let's call them molecular footprints from the mitochondria's past.
Footprints, like clues.
Yeah, clues that resemble bacterial components. And the immune system, which is primed to spot bacteria, sometimes reacts as if there's an infection.
Even though it's just our own mitochondria acting up, or maybe leaking.
Exactly. Think of it like a long-term living arrangement, mostly peaceful. The host cell and mitochondria, they coexist, they benefit each other.
Power for the cell, a safe home for the mitochondria.
Right. But this paper argues that under certain conditions, certain stresses, that arrangement can break down. The host cell starts to see its own mitochondria as foreign.
And that mistaken identity, that's the trigger for inflammation.
That's the hypothesis. When mitochondria are stressed or damaged, they can release these molecules that look bacterial. The immune system detects them, sounds the alarm, and boom, inflammation.
Now, a bit of inflammation is okay, isn't it? For healing and fighting bugs.
Oh, absolutely essential. Acute inflammation is vital. The problem arises when it's the wrong signal or it just doesn't shut off. Chronic inflammation.
And that's linked to all sorts of diseases.
A huge range, yes. Autoimmune diseases, metabolic issues, neurodegenerative conditions.
So this ancient partnership, usually good, can become a source of chronic disease if the communication breaks down.
Wow.
It is a powerful concept.
So what are some of these bacterial leftovers or footprints that trigger the alarm?
Okay, well, there are several key ones they highlight. One big one is mitochondrial DNA, mtDNA.
Different from the DNA in our cell nucleus.
Yes. Structurally, it's often circular, like bacterial DNA, but more importantly, it generally lacks a type of chemical tag called CpG methylation.
Methylation, that's like a little chemical note on the DNA.
Sort of, yeah. Our nuclear DNA has lots of these methyl tags, especially in certain regions. Bacterial DNA typically doesn't, and neither does mtDNA for the most part.
Okay.
So if mtDNA somehow escapes the mitochondrion and gets into the main cell cytoplasm...
Where it shouldn't be.
Exactly.
Yeah.
Immune sensors there, like toll-like receptor 9, TLR9, or another one called cGAS, they spot this unmethylated DNA.
And they think bacteria, or maybe virus. Pretty much. They recognize that pattern as danger or foreign. And activating these sensors kicks off an inflammatory response, including making things like type interferons.
Interferons. I know those are involved in fighting viruses, but also in autoimmune diseases like lupus, right?
Correct. So escaped mtDNA can directly fuel that kind of inflammation.
Wow. So the mitochondria's own genetic material becomes a danger signal. What else? You mentioned peptides.
Right. N-formylated peptides. This is another fascinating bacterial echo.
N-formylated.
Okay. So when bacteria make proteins, the very first amino acid in the chain usually has a little chemical group attached, a formyl group. It's called N-formyl methamine, or fMet.
And our regular cell proteins don't start that way.
No, the proteins made by our ribosomes in the cytoplasm don't have that formal group, but guess what does?
Let me guess. Mitochondria.
The proteins made inside the mitochondria, they still use this fMet start signal, just like bacteria.
That's amazing. A direct link.
It is. And our immune cells, especially phagocytes like neutrophils, have receptors on their surface specifically designed to detect these N-formylated peptides. They're called N-formyl peptide receptors, PRRs.
So they're basically sniffing for bacterial bits.
Exactly. It's a way to detect bacterial invasion or tissue damage where bacteria might be involved. But if mitochondria get damaged and release their proteins,
those fMet peptides can trigger the same alarm.
Precisely. Another case of mistaken identity based on ancient biochemical similarities.
Wild. Okay, DNA, peptides. You also mentioned a specific fat, cardiolipin.
Yes, cardiolipin. It's a type of phospholipid, a fat molecule. It's quite abundant in bacterial membranes.
And mitochondria.
And primarily in the inner mitochondrial membrane in our cells. It's really important there for the structure and function, especially for the energy production machinery.
So normally it's tucked away deep inside.
Right. But under stress or during cell death processes like apoptosis, cardiolipin can actually move to the outer mitochondrial membrane. It gets exposed.
And that exposure is a signal.
It seems to be, especially if the cardiolipin gets oxidized, chemically modified by reactive oxygen species, which mitochondria can produce under stress.
Okay.
This exposed, oxidized cardiolipin can do a couple of things. It can help form pores in the outer membrane, letting other mitochondrial danger signals out.
Like the mtDNA we talked about?
Potentially, yes.
Yeah.
And it might also directly activate parts of the inflammasome, specifically the NLRP3 inflammasome, which is a major driver of inflammation in many diseases.
So a stressed mitochondrion literally waves a bacterial fat flag on its surface.
That's a good way to put it. It signals distress in a way that the cell interprets through these ancient danger-sensing pathways.
Okay, one more you mentioned. Double-stranded RNA. I usually think viruses with that one.
You're right. dsRNA is a classic viral signature that our cells are very alert to. But mitochondria can make it too.
Oh.
It relates to how the mitochondrial DNA is organized. The genes are packed very tightly, sometimes overlapping, and encoded on both strands of the DNA circle.
Ah, okay. So when it's being read, transcribed into RNA.
Exactly. The transcription process can sometimes generate complementary RNA strands from opposite DNA strands in the same region. These can then pair up to form double-stranded RNA.
I see. Accidental dsRNA formation.
It seems so. And if this mitochondrial dsRNA gets out into the cytoplasm...
His cells' viral sensors pick it up?
Yes. Sensors like RIG-I and MDA5, which are part of the RIG-I-like receptor family, they detect this dsRNA, think virus, and trigger another inflammatory pathway, often involving a protein called MAVS, which sits on the mitochondrial outer membrane itself.
So mitochondria can accidentally mimic both bacteria and viruses to the immune system.
It appears so. Through their DNA, their peptides, their lipids, and even their RNA. These are all echoes of their ancient past.
Okay, so putting it all together, we have these essential powerhouses, our mitochondria, still carrying these molecular signatures from their bacterial ancestors. Normally, everything's fine, contained.
Kept under wraps, yeah.
But the paper argues that a break in endosymbiosis, maybe due to stress, damage, whatever, causes these bacterial-like bits to leak out or get exposed.
Right. The containment fails.
And our immune system, using ancient detection systems, sees these mitochondrial parts as foreign invaders, triggering inflammation.
That's the central idea.
And the really big implication is that this mechanism, this breakdown of an ancient truce, might be a key reason why we're seeing more inflammatory diseases today.
That's what Murphy and O'Neill propose. They link it to modern lifestyle factors, things like obesity, which causes metabolic stress, big changes in diet, lots of processed foods, chronic psychological stress, maybe lack of sleep, environmental pollutants too.
All these things putting extra strain on our cells, and specifically on our mitochondria.
That's the suggestion, that this chronic low-level stress disrupts mitochondrial function and integrity, leading to more leakage of these danger signals.
Making the immune system constantly simmer, leading to chronic inflammation.
Exactly, and that could contribute to the rising incidence of diseases like, well, lupus, multiple sclerosis, rheumatoid arthritis, inflammatory bowel disease. The list is unfortunately long.
It connects the dots between our environment, our lifestyle, and this really deep cellular biology, this ancient pact.
It provides a potential underlying mechanism, yes.
And if this understanding holds up, it could change how we treat these conditions.
Absolutely. That's the exciting therapeutic angle. If we know these specific mitochondrial signals and the pathways they trigger are driving disease, maybe we can intervene there.
How? Like shoring up the mitochondria, stopping the leaks.
Could be developing drugs to improve mitochondrial health and stability, or perhaps blocking the release of specific molecules like mtDNA or fMet peptides.
Or maybe blocking the immune receptors that detect them, like TLR9 or the PRRs.
That's another avenue, yes.
Mm-hmm.
Or modulating the downstream inflammatory pathways like the inflammasome or interferon production, but perhaps targeted more towards the mitochondrial triggers. It opens up new possibilities beyond just general immune suppression.
Addressing the root cause, or at least a very deep-seated cause, within the cell itself.
That's the hope. Targeting the breakdown in this ancient endosymbiotic relationship.
It really makes you pause and think, doesn't it? Could this rise in inflammatory diseases really be, at least partly, our modern lives, stressing out this billions of years old partnership inside us?
It's a compelling narrative.
Making our own inner bacteria seem like a threat again. And it makes you wonder what other connections between our deep evolutionary past and our current health are still waiting to be discovered.
That's a million-dollar question, isn't it? There are likely many more layers to uncover about how our history shapes our biology and our susceptibility to disease. It underscores the importance of basic research into these fundamental processes.
Absolutely fascinating. A really thought-provoking deep dive today, connecting inflammation right back to the dawn of complex life. Thank you for explaining all that.
My pleasure. It's a really dynamic field, and this perspective, thinking about that ancient contract, is certainly adding a lot to the conversation about inflammatory disease.
Well, that's all the time we have for today. Please don't forget to like the video and subscribe if you want more content like this.
See you next time.